Thanks for joining us in person today. Those that are here, as it's now 9 a.m., we'll start the broadcast for our attendees. Ladies and gentlemen, I have been informed that there is a quorum present, and it is now just after 9 a.m. I declare this annual general meeting of Imagion Biosystems Limited open and welcome you to this physical and virtual meeting this morning. My name is Bob Prue, CEO and Executive Chairman of Imagion Biosystems, and I would like to thank you for your interest in and your support of the company. I'm again delighted to be here in Australia to join my fellow directors and colleagues to host this meeting as a hybrid forum and to make ourselves available to meet face to face. Our AGM is being held on the lands of the Wurundjeri people, and I wish to acknowledge them as traditional owners. I would also like to pay my respects to their elders, both past and present. I would now like to introduce the other members of the board, Mark Van Asten, Jovanka Namoska, and Diane Angus joining me in Melbourne, and David Ludvigsen and Mike Harsh joining us online. Our CFO and Company Secretary, Jeff Hollis, is also here in Melbourne. I would also like to welcome Billy Chen, a representative of the company's auditor, RSM Australia Partners. Given this is again a hybrid meeting, I just want to run over some housekeeping matters before we commence with the formal business. At this meeting, there will be 11 items of business, including 10 resolutions. Each resolution will be proposed and voted on as a simple majority to be carried, except for item 10, which is resolution 9, and then item 11, which is resolution 10, which are special resolutions. In order for resolutions 9 and 10 to be passed, at least 75% of the votes validly cast by shareholders on the resolutions must be in favor. Today's meeting is being held physically here in Melbourne and online via the Lumi platform. This allows shareholders, proxies, and guests to attend the meeting either physically or virtually. All attendees can watch a live broadcast of the meeting. In addition, shareholders and proxies have the ability to ask questions and submit votes. Online attendees can submit questions at any time. To ask a written question, select the messaging tab at the top of the Lumi platform. Type your question in the box towards the top of the page and press the arrow symbol to send it. A copy of your submitted questions along with any written responses from our meeting team can be viewed by selecting My Messages. To ask your question verbally, pause the broadcast on the Lumi platform and then click on the Request to Speak button. The audio question interface will be displayed. You will be prompted to confirm your name and enter the topic of your question. Submit your request and follow the instructions to allow access to your microphone and connect into the queue. Please note that while you can submit questions starting now, I will not address them until the relevant time in the meeting, when I will also address any questions from those physically in attendance. Please also note that your questions may be moderated, or if we receive multiple questions on one topic, we may combine them together. Finally, due to time constraints, we may run out of time due to all of your questions. If this happens, we will answer them in due course. Voting today will be conducted by way of a poll on all items of business. In order to provide you with enough time to vote, I will shortly open voting for all resolutions. At that time, if you are eligible to vote at this meeting, a new voting tab will appear. Selecting this tab will bring up a list of resolutions and present you with voting options. To cast your vote, simply select one of the options. There's no need to hit a submit or enter button, as the vote is automatically recorded. You do, however, have the ability to change your vote, right up until the time I declare voting closed. I now declare voting open on all items of business. The voting tab will soon appear. Please submit your votes at any time. I will give you a warning before I move to close voting. Before attending to the formal business of the meeting, I'd like to take this opportunity to reflect on our accomplishments of 2022 and provide some insight regarding what we anticipate for the future. As we noted in our annual report, 2022 was a momentous year. We showed that our imaging agent technology was safe and well tolerated, and not just in healthy volunteers like most phase 1 studies, but in actual breast cancer patients. Additionally, we ended this year being the first company ever to conduct a clinical study and present clinical data for the targeted imaging of cancer using magnetic nanoparticles. We've shown that our nanoparticle imaging technology could be used to non-invasively detect cancer, not just in laboratory animals, but actual human patients. I think both from a scientific perspective and a business perspective, this is a major success. Yes, recruitment has been challenging, but we did it. We have positive data from patients with metastatic breast cancer. If you compare our achievement to the number of drugs or devices that fail in early stage clinical studies, we've delivered a significant milestone. And now we've carried the momentum from 2022 into 2023. In early February, we reported that a review of our phase 1 data by an independent group of radiologists corroborated two key findings. First, that our MagSense imaging agent was detectable in the lymph nodes. And second, that by using magnetic resonance imaging, the radiologists observed a differentiated image between normal lymph nodes and nodes that could contain cancer cells. This independent corroboration of the study imaging results has enabled us to shift our strategy for our nanoparticle pipeline towards using MRI for the molecular imaging of cancer. The MRI approach opens the door to a faster and more economical path to commercialization, developing a whole new type of detection technology. And we anticipate that by leveraging existing MRI, we can develop a more proximate revenue path for our imaging agents, initially targeting HER2 breast cancer, and in the future, other cancers such as ovarian and prostate cancer. This year, we will continue to advance our pipeline for molecular MRI, as we believe this strategy to be more attractive for strategic partners and more likely to achieve commercial success sooner for us. By prioritizing molecular MRI for our nanoparticle technology, our development path has become clearer and less complicated. And as a result, we were able to initiate communication with the US FDA to expand our MagSense HER2 development for breast cancer detection to a phase 2 study. Over the next several months, we will be completing additional work and preparing our submission for an investigational new drug or IND application to the FDA. Receiving clearance of an IND is a major technical and regulatory milestone in and of itself. Most biotech and pharma companies see the IND as a significant milestone and value inflection point because the rigor and supporting evidence needed to gain FDA's IND clearance is really important. Since the beginning of this year, we've continued to enroll into the phase 1 study and are expecting soon to complete the second cohort of patients. Given the satisfactory evidence supporting our primary and secondary endpoints and the recent guidance we have received from the FDA regarding our plans for the phase 2 study, we believe we can now begin winding down the phase 1 study. I acknowledge that it's taken longer to complete than we thought when we initiated the study, but the positive outcome of this study marks an important accomplishment for the company. In summary, in the past 2 years, we've taken our lead product for breast cancer from benchtop to a positive outcome in a phase 1 study. Additionally, we've demonstrated that our nanoparticle technology is indeed a platform for targeted molecular imaging, having reported preclinical results for both prostate cancer and ovarian cancer. Pursuing use of our nanoparticle technology with conventional MRI will reduce the risk, time, and cost of our development path. Additionally, it is more likely to be an attractive commercial prospect for partners while still preserving the opportunity for our proprietary magnetic relaxometry technology to play a role in the future. Okay, with the success we are claiming from our phase 1 study, you may be asking why we need to do a phase 2 study or what we expect to gain from it. Firstly, I'd like to remind everyone that the phase 1 study was always intended as a proof of principle for our nanoparticle technology. Since our nanoparticles had never been tested in human subjects before, it was important that we start with a limited study before committing to larger numbers. And because no one had ever tested a targeted imaging agent before, and because we weren't only testing for safety in healthy volunteers, in our phase 1 study, we had to take our best guess as to what dose to give patients and what time points or points we should be imaging. But by including this as part of our phase 1 study, we now have data to inform the next study. As challenging as it's been, these first patients have been instrumental in establishing the baseline for our future work. In the phase 2 study, we will start with dose ranging, that is, reducing the dose to see how low we can go while still achieving detectability. Think of this for a minute. This is good all around. Patient safety is likely to be better with smaller doses, and our gross margins can be higher if we use a smaller amount of imaging agent to achieve the same diagnostic outcome and price. Additionally, in the first patients of the phase 2 study, we will be looking to optimize the timing of when the MRI is taken. Once we've established a more optimal dose and timing, we will expand the study to larger groups of patients. Okay, given how the slow recruitment in phase 1 study here in Australia, what does that mean for recruiting larger numbers of patients in phase 2? There are three things working in our favor. Firstly, in many countries, including the US, the population size means that there's just a lot more patients will be available to us. Secondly, in larger numbers of jurisdictions, we have many more sites we can use to recruit in parallel. And thirdly, since we were only going to test a limited number of patients in phase 1, we had to focus on patients suspected of nodal disease. This has meant that for the phase 1 study, more than half of the HER2 breast cancer patients have not been eligible. So in the phase 2 study, we'll be able to recruit from all HER2 positive patients, giving us a larger patient population. In 2022, we made real progress in our mission to improve cancer detection and have chartered a clear path with an updated strategy that will move us towards our goal of commercializing our nanoparticle platform. I understand that for our shareholders, it can be hard to be enthusiastic about these wins or accomplishments and the prospects of the future when our share price is suffering. And the fact that over the last year, approximately 80% of the ASX biotechs have been trading down is no real consolation. Recently, I heard of an analyst who's been watching IBX. He told a colleague that he liked the IBX story, but that we still had some wood to chop. I liked this analogy because it implies if we do the work, we have the makings of a roaring fire. In my opinion, and that of our board, the IBX share price does not reflect the value we've created and are going to be creating. Over this past year, we've seen a number of the large pharma companies, including Novartis, Merck, and Bayer, being interested in the diagnostic imaging space and licensing and partnering with companies developing targeted radio tracers and radiopharmaceuticals. We believe this bodes well for our strategic plan of partnering. And towards that end, we've recently opened a secure data room where we can share confidential information with prospective strategic partners. With our MagSense imaging agent for breast cancer now laying the groundwork for our technology to be first in class for molecular imaging by MRI, we believe we can change the way we see cancer. To realize this potential, we have to continue to push forward. Every additional step through the clinical and regulatory process adds value and takes risk off the table. But it also requires having access to working capital. We know that markets and economic conditions in Australia, and indeed globally, have been difficult and may continue to be so. So I'd particularly like to thank the shareholders that participated in the recent entitlement offer. Your support for the company is greatly appreciated. It is exactly those market conditions that also highlight why having a financing instrument like we have put in place with Mercer Street Capital is so important for a company like Imagion. We don't intend to solely rely on this instrument to fund the company, but having it means we have access to working capital without having to worry about timing of market conditions. And with $3.5 million of funds expected to come in this quarter from our ATO tax credit, we aim to use the Mercer funding judiciously to keep our drive towards the IND on track. Now, before closing my remarks and moving on to the formal meeting, I'd like to speak briefly to the recent announcement of my plans to retire from the role of CEO. While I'm not the founder of the company, I fully vested myself in the vision and mission of Imagion and believe strongly in what we are trying to accomplish and the value it will create. So I can assure our shareholders that my plans to retire from the role of CEO does not mean I'm going to be riding off into the sunset. The board and I are fully committed to the principles of good governance and plan for me to remain as chairman of the board to keep the momentum going and ensure we preserve the knowledge and history gained over the years of my leadership so that we are successful in the next phase of our journey. In closing, I'd like to thank all of the Imagion stakeholders for their support. Shareholders, customers, collaborators, advisors, employees, directors, and particularly those breast cancer patients that have, despite their medical diagnosis, participated in our study, and the physicians and study teams who executed on the protocol. We are looking forward to our future and to achieving our goal of changing the way we see cancer to positively impact patient outcomes. I'll now move on to the formal business of the meeting. Item 1, financial and related reports. This item requires us to receive and to consider the financial report of the company and its controlled entities and the related directors and auditors' reports in respect of the financial year end 31 December 2022. There is no requirement for shareholders to vote on these reports. Are there any questions or comments on the report from the room? If not, I'll ask Mr. Hollis if there's any further questions or comments that have been submitted in relation to item 1 to the management or auditors of the company.
No questions.
No questions? Okay. The next item of business require a shareholder vote and will be conducted via an online poll as previously mentioned. Item 2 is to consider and, if thought fit, to pass the following resolution as a non-binding ordinary resolution. That the remuneration report of the company and its controlled entities for the year ended 31 December 2022 be adopted. Please note that a vote on this resolution is advisory only and does not bind the directors of the company. The board abstains from making a recommendation in relation to this resolution. There is a voting exclusion on this resolution. The key management personnel, as set out in the company's 2022 annual report, and their closely related parties must not vote on this resolution. Any exception to this prohibition exists where the person is exercising a directed proxy, which specifies how the proxy is to vote on the proposed resolution on behalf of someone other than the key management personnel or the closely related party. A key management personnel or their closely related party that is appointed as a proxy must not exercise the proxy on a resolution connected directly or indirectly with the remuneration of a key management personnel if the proxy is undirected. That is, if the appointment does not specify the way the proxy is to vote on the resolution. However, the prohibition will not apply if the person is the chairman of the meeting at which the resolution is voted on, and the shareholder expressly authorizes the chairman to exercise the proxy, even if the resolution is connected directly or indirectly with the remuneration of the key management personnel of the company. I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions or comments in relation to item 2 from the room? Mr. Hollis, any further questions come in?
No, there aren't.
No questions. I'll now remind you to register your online vote either for or against in relation to item 2 concerning the approval of the company's remuneration report. Item 3 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That Mr. David Ludvigsen, who retires in accordance with clause 20.3 of the company's constitution and ASX listing rule 14.5 and being eligible, be re-elected as a director of the company. The directors of the company, other than Mr. Ludvigsen, recommend the shareholders vote in favor of this resolution. And I report that the valid proxies received in advance of the meeting are displayed on the screen. Any questions or comments in the room related to item 3? Any questions coming in online? No. Okay, with that, we'll move on to item 4. Item 4 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That Ms. Diane Angus, who retires in accordance with clause 20.3 of the company's constitution and ASX listing rule 14.5, be eligible, be re-elected, and being eligible, be re-elected as a director of the company. The directors of the company, other than Ms. Angus, recommend that shareholders vote in favor of this resolution. And I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions in the room related to item 4? Any coming in online? Thank you. We'll move to item 5. Item 5 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That for the purposes of ASX listing rule 7.2, exception 13, and for all other purposes, the shareholders approve the company's employee incentive plan and for the issue of securities under that plan on the terms and conditions described in the explanatory memorandum accompanying this notice. There is a voting exclusion on this resolution. Any person or their associates who are eligible to participate in the employee incentive plan must not vote on this resolution. The directors of the company recommend that shareholders vote in favor of this resolution. I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions in the room from item 5? Any coming in online? None. I now remind you to register your vote, your online vote, either for or against in relation to item 5 concerning the new employee incentive plan. Item 6 is resolution 5, the ratification of prior issue of commencement shares, first tranche convertible notes, and first tranche options A to Mercer Street Global Opportunity Fund. Item 6 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That for the purposes of ASX listing rule 7.4 and for all other purposes, the issue of 22,058,824 shares, 1,650,000 convertible notes, and 14,138,956 options in the company to Mercer Street Global Opportunity Fund, LLC, which issues occurred on 21 March 2023 on the terms and conditions more particularly described in the explanatory memorandum accompanying this notice, that it's ratified. There is a voting exclusion on this resolution. Mercer Street Global Opportunity Fund or their associates must not vote on this resolution. The directors of the company recommend that the shareholders vote in favor of this resolution. And I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions in the room related to item 6? Any online? Online. I now remind you to register your online vote either for or against in relation to item 6 concerning the ratification of prior issue of commencement shares, first tranche convertible notes, and first tranche options A to Mercer Street Global Opportunity Fund. Item 7. Item 7 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That for the purposes of ASX Listing Rule 7.1 and for all other purposes, approval is given for the issue of 27,995,876 options in the company to Mercer Street Global Opportunity Fund, LLC on the terms and conditions more particularly described in the explanatory memorandum accompanying this notice. There is a voting exclusion on this resolution whereby Mercer Street Global Opportunity Fund or their associates must not vote on this resolution. The directors of the company recommend that the shareholders vote in favor of this resolution. And I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions in the room related to item 7? Any online? Nope. Okay, again, I remind you to register your online vote either for or against in relation to item 7 concerning the approval of the future issue of first options B to Mercer Street Global Opportunity Fund. We'll move to item 8. Item 8 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That for the purposes of ASX Listing Rule 7.1 and for all other purposes, approval is given for the issue of 1,100,000 convertible notes and 28,089,888 options in the company to Mercer Street Global Opportunity Fund, LLC on the terms and conditions more particularly described in the explanatory memorandum accompanying this notice. There is a voting exclusion on this resolution whereby Mercer Street Global Opportunity Fund or their associates must not vote on this resolution, and the directors of the company recommend the shareholders vote in favor of this resolution. I report that the valid proxies received in advance of the meeting are displayed on the screen. And are there any questions on item 8 from within the room or online? No questions. I remind you to register your online vote either for or against in relation to item 8 concerning the approval of the future issue of second tranche convertible notes and second tranche options to Mercer Street Global Opportunity Fund. Item 9. Item 9 is to consider and, if thought fit, to pass the following resolution as an ordinary resolution. That for the purposes of ASX Listing Rule 7.1 and for all other purposes, approval is given to the issue of up to 13,750,000 convertible notes and up to 750 million options in the company to Mercer Street Global Opportunity Fund, LLC on the terms and conditions more particularly described in the explanatory memorandum accompanying this notice. There is a voting exclusion on this resolution. Mercer Street Global Opportunity Fund or their associates must not vote on this resolution. I confirm that pursuant to section 9.3 of the explanatory memorandum included with the notice of meeting, that should this resolution be passed, then the approval granted will be valid for a period of 3 months. The directors of the company recommend that shareholders vote in favor of this resolution, and I report that the valid proxies received in advance of this meeting are displayed on the screen. Are there any comments or questions from the room on item 9 or from online? Okay. I now remind you to register your online vote either for or against in relation to item 9 concerning the approval of the future issue of subsequent tranche convertible notes and subsequent tranche options to Mercer Street Global Opportunity Fund. Item 10 is to consider and, if thought fit, to pass the following resolution as a special resolution. That for the purposes of section 136, subsection 2 of the Corporations Act, and for all other purposes, the Constitution set out in Annexure A to this notice is adopted as the new Constitution of the company in substitution for its current Constitution, which is repealed, with effect on and from the conclusion of this meeting, as more particularly described in the explanatory memorandum accompanying this notice. Item 10 is a special resolution. Accordingly, at least 75% of votes cast by shareholders present and eligible to vote in person or by proxy at the meeting must be in favor of this resolution for it to be passed. The directors of the company recommend that shareholders vote in favor of this resolution. And I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions or comments in relation to item 10 from the room or from online? No questions. I now remind you to register your online vote either for or against in relation to item 10 concerning the adoption of the new Constitution. Item 11 is to consider and, if thought fit, to pass the following resolution as a special resolution. That for the purposes of ASX Listing Rule 7.1A, and for all other purposes, approval is given for the company to issue up to an additional 10% of its issued equity securities by way of placements over a 12-month period on such terms and conditions more particularly described in the explanatory memorandum accompanying this notice. Item 11 is a special resolution. Accordingly, at least 75% of votes cast by shareholders present and eligible to vote in person or by proxy at the meeting must be in favor of this resolution for it to be passed. There's a voting exclusion on this resolution. Any persons who are expected to participate in or who will obtain a material benefit as a result of a proposed issue of shares under the 10% placement capacity, accept the benefits solely in the capacity of a shareholder as a holder of shares, and associates of those persons must not vote on this resolution. The directors of the company recommend that shareholders vote in favor of this resolution, and I report that the valid proxies received in advance of the meeting are displayed on the screen. Are there any questions from the room on item 11 or from online? No. That brings us to the end of our formal business meeting. I now ask you to complete your voting papers and to place it in one of the ballot boxes. For those joining us virtually, please note that online voting will remain open for a further 2 minutes and then I will formally close the meeting. The results of the poll will be notified to the ASX and put on the company's website later today. Are there any further questions from those in the room? Any further questions have been submitted online?
We do, yeah. We have a handful of questions from a few of our shareholders, which is great. So a couple from Mr. Michael Dobrojevic. So apologies, Michael, if you're online and I've mispronounced that. With the most recent capital raise, is there any progress on covering the shortfall? So I can probably field that to start with. Yeah, the shortfall was priced at 1.7 cents, and given where our share price is currently trading at 1.5 cents, it is just not feasible at the moment to be placing that at a premium to our share price. We do have until early July to get that done. So we will leave it open for a little bit and we'll make a call on where we think that's heading in the next few weeks and inform shareholders. We certainly did have some good progress prior to our share price falling below the offer price, and those people are still potentially keen, and we're re-engaging with them regularly, but it really is a factor of our share price at the moment. Anything else to add?
No.
Another question from Mr. Dobrojevic. This is again around capital raising. Should future capital raises be targeted only to sophisticated investors who are potentially more likely to support the company long term? this is in Michael's wording here, it seems that the current shareholder base do not have alignment with the positive path for the company. I won't comment on that. That is everyone else's, that's their prerogative. But we decided this time around, instead of doing a placement to sophisticated and high net worth investors, that we would do an entitlement offer to give everyone a chance on the register to participate. And we're very grateful for, we raised over $400,000 in this climate with where our share price was at. We're very grateful for that and really appreciate that support. Whilst we also thought we would have a shortfall of $1.5 to $2 million, that we were looking at sophisticated investors and high net worths. So we were kind of trying to appease everyone. Unfortunately, given market conditions and where we're at, it didn't quite come to fruition, but we certainly don't regret going down that path. Another question from Mr. Dobrojevic. In the most recent annual report, nearly half of the shareholders have holdings less than a marketable parcel. Are there any plans to buy these shares back? So just a marketable parcel is less than $500 worth of shares, just to spell that out. Two answers to that. One, it is quite you've got to pay for the shares. It's around 40 million shares at our current share price, or maybe closer to 50 at the moment. If our share price was 5 cents, for example, it'd be a far less amount of shares. So a lot of these shareholders have bought very small parcels of shares. And for us and the business we're in, I would really, as much as sometimes it's a pain handling 8,500 shareholders and you have to mail out AGM notices and the like, a lot of these investors have invested for a reason that, one, they want to see cancer detected earlier, and it's very much an emotional journey for them. So I don't believe that going down the route of buying back those shares, even though we're entitled to, is something that we're considering at the moment. Anything else to add on those? So thanks, Michael, for those questions. A few questions from Mr. Anestis Lazaridis.
So do we want to close voting before we keep going?
Oh, we can. Yeah, it's been a couple of minutes.
Yeah, it's been a couple of minutes. So we can, at this point in time, we'll declare voting closed and we'll continue with the Q&A session.
Yep. I'll direct these through you, Bob, and anyone else, feel free to jump in. So sort of two-parter here. What strategies are being or going to be employed to recruit patients? And you coincided with patient numbers. How was and is the sample size determined for the phase 1 and phase 2 study?
Yeah, so I'll answer those in reverse order. Going into the phase 1 study, we really didn't have to worry too much about the quantum because that's pretty much dictated by the regulators. When you're doing an early phase study like that, they pretty much restrict how many patients you can get access to, and then they want to see the data before they're going to give you permission to go more. So we knew going into it that the target of somewhere between 10 and 15 was likely to be where we would start. We designed that study with being able to go up to 30 because we didn't know what kind of data we would get. But the idea was that we'd start with a simple phase 1 study of somewhere in the order of 10 to 15 patients. As you go into the next phase of things, now you start to worry about the statistical relevance of what you're looking at. And so the number of patients that we will plan to recruit into the phase 2 study will be driven by the biostatisticians looking at, well, what are the endpoints we're trying to achieve? And how many patients do you have to have in order to be able to be confident in your data supporting that? So we're just in the process of doing that now. Part of the run up to the IND filing is in fact getting the biostatisticians involved with the clinical investigators to identify the endpoint or endpoints so we can then back calculate how many patients do we think that we're going to need to be able to get to that proof of evidence. And one of the things that's important there is, we were discussing this the other day, is as you go through this process of going from phase 1 to phase 2 to phase 3, the ability for the sponsor, i.e. the company, to be involved as the study is going on gets less and less. So what that means is that by the time we're doing a phase 3 study, we have to have defined the exact protocol, exactly what's going to be done for every patient, and what our endpoints are, because we can't really change it in the middle of the study. So you're committed to something, you're actually blinded to the data until the end. And so if you don't get your study design right, you've tested a whole bunch of patients, And at the end, you find out, did you achieve what you wanted to achieve or not? So the reason that we're doing a phase 2 instead of going all the way to a phase 3 is we still have some flexibility in the phase 2 to try to figure out exactly what those endpoints we want are, so that when we do go spend the money on the larger study, we're pretty darn sure that we're going to get the outcome that we wanted to, because we really need to make sure that we've locked everything down. So the trick right now is, in the next couple of months, to work with our investigators and the biostatisticians to say, if these are the endpoints that we want to try to get to in phase 2, so that we know what we're planning for phase 3, how many patients do we think we need to do? And so we'll be in the process of doing that. And that will then drive, for example, how many study sites do we need, and how quickly do we think that we can recruit patients into that study? One of the things I mentioned in my chairman's remarks was there are a few things working in our favor now for phase 2 compared to phase 1. One is we can have a lot more study sites. We're planning to have, probably more than 10 study sites, whereas in phase 1 we only had 4 here in Australia. In Australia, we have 25 million people, of which HER2 breast cancer is only 20% of breast cancer. So our patient population was immediately very restricted. In the United States, we're going to have 350 million people, but with the same incidence of breast cancer. So the numbers are just bigger. And so we know, for example, that hospitals in the United States have typically in the order of 5 to 10 times the number of patients with breast cancer that you might see at a hospital here. So the trick will now be for us, again, same thing, over the next 6 months or 3 months while we're working towards our IND, is do the analysis of how many sites do we need based on the statistics of what we have to achieve, and what are the sites telling us in terms of the number of breast cancer patients that they see on a regular basis that we could potentially enroll. And then again, one last thing working in our favor is in the phase 1 study, because we really wanted to know whether or not we see cancer in lymph nodes. It wouldn't have done us any good to recruit patients that were node negative. We'd find out whether our nanoparticles get into the lymph nodes, but we wouldn't know whether or not we were detecting cancer, because those patients had, by definition, a priori, didn't have nodal disease. So we restricted our phase 1 study to only be patients that were expected to be node positive. Node negative patients are more than half of breast cancer patients. So we automatically started that study restricting half of the patient population. So when we go into the phase 2 study now, in order to prove our case that we can differentiate node positive from node negative, we're going to have to recruit all breast cancer patients to see if we get the answer. So the number of patients available to us by population, the number of patients available to us by number of study sites, and the number of patients available to us by eligibility will all be increasing, which is why we think we'll be able to get through the phase 2 study in a reasonable period of time. But exactly how much time will depend on how many patients and how many sites we have. And we're just starting to accumulate that data now. So I can't quite get an answer today on how long that's going to take, but we'll be working on that as we move towards the IND filing.
And we'll give some updates and indications.
Absolutely. As it starts to unfold and we get clarity and we have that trajectory planned out, we'll be able to share that.
Yeah, I guess sharing something now would be a guess. And we know what happens in the minds of shareholders when we put out information that's not accurate. So, are there plans for collaboration with other research groups or institutions in the future? Maybe just touch on where our MassGen and
So while we're very focused, the majority of our effort is focused on this HER2 breast cancer, getting it through the clinical phases. I'd say there's 2 other aspects to our research programs. One is our immediate pipeline of other potential cancer imaging targets like prostate cancer and ovarian cancer. So we have some collaborations right now in prostate cancer. They're relatively small while we're doing early stage work, but those collaborations are likely to grow as we start to focus on the clinical utility for prostate cancer. The same thing for ovarian cancer. We'll have to find those researchers that are interested to work with us on the clinical development of ovarian cancer. Behind that, and we have, a relatively small project working with the University of Sydney in the brain cancer that we have. Behind that is some fundamental R&D work that goes on. We're constantly looking at other utility for our particles. And so we have collaborations looking at things, everything from how our particles might be used from a therapeutic perspective to collaborations on what other targeting ligands or moieties we might use to target different types of cancer. So they tend to be relatively low level, from an expenditure perspective, because they're early stage work in that, but we're always looking at additional collaborations that can support ultimately getting us into the clinic for our products. Because from our perspective, we're not interested in being just a fundamental R&D organization. Our goal is really to drive getting products into the clinic so that they can ultimately be approved by the regulators for actual use in human testing.
Yeah, any follow-ups?
No, it's not.
Got some questions through from Dr. David Butterfant, another holder. This one's back to the phase 1. What has been the rationale behind not incentivizing patients as it is a non-invasive procedure?
Yeah, so two things there. One is incentive for people to participate in phase 1 is usually associated with what's known as a healthy volunteer study. That is, we're simply trying to identify for a drug whether or not, if this drug is administered to someone who doesn't have a disease, is a healthy individual, whether there's any side effects, toxicity problems, or anything. And so you're asking people who are otherwise perfectly healthy to be given a dose of a drug that they've never seen before. And so it's very common in that scenario to provide a financial incentive to participate in those studies. It's not seen as being unethical because you're asking somebody who would otherwise not need any kind of treatment to be given something. When you move away from a healthy volunteer study to the kind of study we did, where we're actually looking at actual patients, in our case, HER2 positive breast cancer patients, the ethics associated with incentivizing people to participate starts to get a little bit fuzzier, right? Because now these patients are considered, or these subjects for the study are considered at risk or vulnerable, right? If you incentivize them to participate in your study, are you disproportionately weighing a financial benefit to them compared to just the standard of care? These are people who have been diagnosed with a disease, and you want to make sure that you're not interfering with their patient treatment and the standard of care. And there's always a concern that if you try to incentivize them financially, that they will make a decision that is more based on the finances than it is on their health care. And so it's pretty much seen as being the ethical boundary that when you're recruiting patients into a study that are actual patients with the known disease, that you don't add a significant financial incentive for them to do that. Now, you can add compensation, which we do. If someone needs to stay in a hotel, or if they don't live proximal to where the study site is, you can pay for transportation. You can compensate them if they miss work. You can compensate them at normal labor rates for them so that they're not out of pocket, having participated in your study and having lost wages. So those things can be done, and that's all considered fair and ethical, and we include that in our study. But it is typically not seen as being ethical to try to financially incentivize somebody to make a decision to do something that may or may not be in their best standard of care interest.
Thanks, Bob. A few other questions. Are there plans, just sort of a two-parter, are there plans to publish the results of the phase 1 study in a scientific journal, and potentially when could that happen? And what are the predefined benchmarks or standards against which the results will be compared to?
Okay, so I'm going to reverse those two. So in our study, the standard of care, or the benchmark, is the clinical evaluation that is given to the patient before we do our study, and the pathology that is assessed of the actual tissue that's excised from the patient. So in our study, a physician, the doctor, assesses the patient. They have been identified by doing a biopsy of the primary tumor that they're HER2 positive. And now the evaluation is typically by ultrasound to look and see whether or not there's the nodes appear to be involved. And then, depending on how the ultrasound looks, they may then do a biopsy of one or more nodes, and they take that tissue and they look in the petri dish and they look for the pathology to see if there's cancer cells there. So the normal clinical evaluation is doctor's evaluation and an ultrasound. And so our first comparison is What did the doctor say? Is this patient node positive or node negative by clinical evaluation? And then what do we say with our imaging results, and how does that compare to what the actual biopsied sample looks like? So we have a very good way to compare our results to standard of care and the clinical evaluation. And so then the first part of that question was
Publishing results.
Publishing results. So right now we're in the process of starting to wind down the phase 1 study. Well, then, you guys very familiar with this. You then, it takes a while for all the data to be compiled. You got to make sure that all the data in the database is locked down, et cetera. But once that data are there, I fully expect that our team, in concert with the study investigators, will publish. I mean, one of the reasons that the investigators like to participate in the study, they're typically academics, they're at hospitals and that. And so they like to, for them, it's a benefit to participate in the study, not only to help their patients, but they get the publication out of it as well. So we would fully expect to do that. I would say, it's probably going to be not less than 6 months, though. It takes a long time for the database to get locked down and all the data to be reviewed. And then if you're going to submit into a scientific journal, they're peer-reviewed journals, and you go through multiple iterations of where this other scientists read your results and ask questions and ask you to comment on things. So it's not something that's going to happen quickly, which is why we felt it was important to at least publish the poster that we presented at the San Antonio Breast Cancer Conference, because at least then our shareholders get to see some portion of the data. But we'll plan to do a more thorough publication, but it does take time to get through the scientific rigor.
Like anything in biotech, it takes time. Just another question from Dr. Butterfant. What limitations and constants and constraints in terms of resources, funding, or technology that may affect the scope of future studies like the phase 2?
So by having moved to MRI for our detection platform, we actually reduced that burden. Had we continued with development of the magnetic relaxometry technology, we would have had both a financial and technical constraint of having to make instruments that would be put in the hospitals for the testing. By moving to MRI, we're taking advantage of the existing infrastructure. Almost every hospital that we would be interested, or clinic that we would be interested in working with, will have a commercially available clinical MRI machine. So that actually reduces the financial and technical constraint. There is nothing, I want to be careful here, there's nothing special about, there's something special about our nanoparticles as an imaging agent, but there's nothing special about the imaging equipment itself. We don't have to have a special version of software running on these MRI machines. We don't have to have a special coil to do the pulsing of that, right? So all of the hospitals that we're going to be working with, and it can come from any vendor, GE, Siemens, United Imaging, Philips, all the major vendors of MRI machines, their machines will work perfectly well with our imaging agent. So the technical constraint is a lot less by having moved to the MRI detection. The financial constraint will be largely driven by how many patients, how many clinical sites to appropriately fund the study.
Yeah, one more question. What are the long-term implications, I guess, of our technology for future research or clinical applications? And what are those potential applications, I guess, beyond the phase 1 study?
Yeah. So I think they're here. The thing that's right in front of our face is that we can now start to do what's known as molecular imaging by the use of our particles. So instead of administering a contrast agent that just helps make the images brighter, gives an improvement in signal to noise, our particles actually generate a contrast that's associated with the fact that we know that the cancer is there because our particles are attaching to the tumor cells. That changes the way the radiologists start to think about, and the physicians start to think about how they could use imaging to make decisions about A, diagnosis, and B, potential treatment. One example in our phase 1 study that we originally started the idea that all we want to do, let's start simple. Can we simply, yes or no, determine whether there are cancer cells in that patient's lymph node? By moving and starting to get images by MRI, what we're noticing is that the initial assessment today by a clinician is, as I said, ultrasound. They're going to take a patient, they're going to do an ultrasound and see if they see nodes that are abnormal in size and shape. If they're abnormal in size and shape, it means that it's probably not a healthy normal lymph node, that node is involved with tumor cells that's caused the change in size and shape. So it's just an ultrasound image looking at morphology or anatomical changes in the lymph nodes. When they look, and typically an ultrasound it can only look at a couple of nodes. They'll look at the nodes most proximal to where the tumor was. What we're finding with the use of our imaging agent is not only can I look at size and shape by MRI, I can look at abnormality of size and shape of the nodes, but I now see whether our particles are generating contrast related to the presence of the cancer cells. So I add one more piece of, let's say, factual data to the radiologist's interpretation. I've got a node that looks abnormal in size and shape, and I see the contrast associated with the particles. Or I see a node that maybe I didn't think was abnormal in size and shape, but I see the particles there. So what I have otherwise maybe thought that node was normal, but in fact, it turns out that there's cancer cells there. But another big benefit that we're seeing is that with MRI, we can actually image a larger number of nodes. And the number of nodes that have cancer cells in it can affect the way the doctor thinks about the course of treatment. It's not just whether or not maybe that patient has one node that's metastatic, but if I've got 5 nodes and I see it's disseminated deeply down into the axillary region, I might think about whether or not I don't want to treat only by neoadjuvant therapy, but maybe I want to do surgical because I want to get rid of as much of that as possible. So what we're already seeing with the use of our technology is we might be able to start to change some aspects of clinical practice because we're giving them more information than today they might have. The next big thing to go after that is, we've said this all along, if we start to demonstrate that we have particles there and they're associated with cancer cells, we know we have the possibility then of that using those nanoparticles as a drug delivery mechanism. If they're there and I'm detecting them, how can I then potentially include a therapeutic payload and start to treat the tumor? So our long-term vision of the evolution of our nanoparticle technology is we affect clinical practice to give the radiologists and the doctors more information to make better decisions about how to treat a patient. But ultimately, we might also play a role in actually treating the patient with our particles.
I confirm no further questions.
No further questions? With that, last chance in the room? Great. I want to thank everyone for attending the meeting. Look forward to the next 12 months and keeping everyone apprised.
Thank you.